Hi everyone, I am Patrice Jane Alhudaya. Let's talk about metabolic disorders. What are metabolic disorders? Metabolism describes the body's ability to convert food and drink into energy. It's a complex process that uses many different chemicals, hormones, cells, and organs to work properly. A metabolic disorder is when something disrupts this process. So there are several different types of metabolic disorders. However, a disorder can be caused when the body doesn't have the right enzyme to break down certain types of foods like fats, proteins, or carbohydrates. An enzyme is a substance that helps a chemical process happen, like breaking down food into something the body can use. Next is a process that helps turns food into energy isn't working properly. For example, sometimes the mitochondria of the cells can't use sugars to make energy like they are supposed to. So, the cells don't get the energy they need and the sugars and oxygen build up and stay in the body. Then, one of the organs involved in the metabolism is diseased or damaged. It could be the pancreas, liver, endocrine glands, or the organs that release hormones, or another organ involved in metabolism. Then lastly is when the body isn't getting enough of a nutrient or mineral it needs to make metabolism work. Many metabolic disorders are genetic or passed down through families or autoimmune, or when body tissues are attacked by its own immune system. Newborns are screened for several serious conditions, including metabolic, hormone, and blood disorders. Early detection and treatment of certain disorders can prevent lifelong health problems. Some metabolic disorders develop during a person's life. The most common is diabetes mellitus. Type 1 diabetes is an autoimmune problem. The body destroys the cells in the pancreas that make insulin. Insulin helps the body use glucose or sugar in the body for energy. Type 2 diabetes is when the body makes insulin but can't use it well anymore. Another is phenylketonuria. It is an example of an inherited metabolic disorder characterized by an inability to break down one of the building blocks of protein, the amino acid phenylalanine. An example of a metabolic disorder affecting fat metabolism is Gaucher's disease, which is characterized by a lack of the enzyme glucocerebrosidase. Metabolic disorders can also be complications of severe diseases or conditions, including liver or respiratory failure, cancer, chronic obstructive pulmonary disease or COPD, includes emphysema and chronic bronchitis, and HIV or AIDS. So what are some treatments for metabolic disorders? Treatment for metabolic disorders depends on the specific disorder. However, common treatments include changes to diet and lifestyle. You may have to avoid certain foods that the body can't handle and break down. Next is medicines. Medicine can be used to give the body what it doesn't have enough of, like insulin in the case of diabetes. Medicines can also be used to control certain symptoms, help you feel better, and avoid life-threatening emergencies. Now, for the prevention, many metabolic disorders can't be prevented because they are genetic or the cause is unknown. But there are things you can do to lower your risk for developing type 2 diabetes, which is the most common metabolic disorder. These include stay at a healthy weight, eat healthy foods like vegetables, whole grains, beans, fruits, and lean meats, stay active or move every day, and drink water instead of juice, soda, or other drinks with sugar.
Good afternoon, everyone. By the way, I'm Erica Tonzo. So for today's presentation, I'm going to discuss about the urea cycle. So urea cycle is also known as the Krebs-Henseleit cycle. It is a very important biochemical pathway whose main goal is to eliminate ammonia from the body by converting it into urea. So first, let's start with an overview. So when you ingest protein or when you break down protein in your body, you of course breaking down amino acids because amino acids are the protein's building block of everything that we put together in our body. When amino acids get catabolized or broken down, they form ammonia. Normally, ammonia is not a problem for a healthy body, but a problem only occurs when you can't get ammonia outside of your body because ammonia basically builds up to toxic levels. So now ammonia is sent to the liver where it will be turned into urea. The urea is then sent to the kidney and eliminated from the body. Remember, only the kidney can eliminate urea from the body. Now everything you see here is the simple explanation of urea cycle wherein it specifically takes ammonia, sets it through a series of very specialized chemical reactions into the liver to form urea, and then takes that urea and ships it to the kidney wherein the kidney can urinate it out. So now let's start with the beginning of the urea cycle. You start with ammonia. Ammonia is formed as a byproduct of amino acids or protein breakdown. Ammonia is combined with carbon dioxide and forms carbamoyl phosphate. The enzyme that catalyzes this conversion is the carbamoyl phosphate synthetase 1. And now, the carbamoyl phosphate then combines with ornithine to form citrulline. That reaction is catalyzed by ornithine transcarbamylase. Remember, everything you see here is not only occurring in the liver, but specifically it occurs in the mitochondria, and the rest of the urea cycle will occur in the cytoplasm. So now that we form citrulline, the citrulline will now combine with aspartate and ATP to form argininosuccinate. The enzyme that catalyzes this conversion is the argininosuccinate synthetase. Now, the argininosuccinate will go on to form arginine, and in the process, it will spit off a fumarate as a byproduct. The enzymes that catalyzes this conversion is called the argininosuccinase. So now, this is the important step in urea cycle, wherein arginine will go back to ornithine, but in the process, it will convert into urea. The enzyme that catalyzes this conversion is known as arginase. Finally, we've completed the major goal of urea cycle. We started with ammonia, the NH3, and we've sent it through a series of reactions, and in the process, we've packaged the ammonia into urea. The urea will then go to the kidney and be exerted in the form of urine. In a normal, healthy individual, this is exactly what must occur in order to get that ammonia outside of the body.
Michael Dominic Signates, and today let us tackle about the urea cycle disorder. A urea cycle disorder, or UCD, is an inherited disease that affects how the body removes the waste that is made from breaking down protein. Everyone needs protein, which is found in food like dairy products, meat, and fish. When a person eats food that contains protein, the body breaks into amino acids, or the building blocks of protein that are used by the body for growth and tissue repair. It changes the rest into nitrogen, which must then be removed by the body. In a healthy person, the liver supplies enzyme to change nitrogen into urea, which is then removed from the body in urine. This entire process is called the urea cycle. The liver in a person with urea cycle disorder is missing an enzyme necessary to convert nitrogen into urea. As a result, ammonia, a highly toxic substance, builds up in the bloodstream and is not removed from the body Untreated, the high amounts of ammonia can cause brain damage, coma, and eventually death.
Next is the causes of UCD or the urea cycle disorder, which is the lack of enzyme or the missing enzyme. Feel free to read below the causes of UCD.
The process of urea cycle disorder are genetic. Genes give the body instructions on how to break down proteins. We usually have two copies of each gene, and most UCD only occur when a person inherits a change gene from both parents. The exception is OTC deficiency, which is passed to the baby through the mother, who is most often not affected. If both parents carry the defective gene, there is first a 25% chance that their children will develop the disorder. Second, a 50% chance that their child will receive one defective gene from one of the parents, which means the child will not show symptoms of the disorder. The child is a carrier. Third, a 25% chance their child will receive both working genes, one from each parent, and will be unaffected. Now, let's go deeper to our discussion. Signs and symptoms of UCD. The age of onset and specific symptoms vary between the different types of UCD. This disorder is most often diagnosed in infancy, but some children do not show symptoms until early childhood. In children with severe UCD, the symptoms will develop within the first 24 hours of life, while all of the symptoms may not be present. Usually, the baby will become very sleepy and irritable and will have feeding problem, including poor, poor feeding, vomiting, and also seizure. Some of the children also experience trouble breathing, and eventually it can cause coma and death. Symptoms in children with mild or moderate UCD who do not show symptoms until early childhood may include these following: disliking meat or other food that is rich in protein, vomiting, and also nausea. Then, mental confusion or hyperactive behavior. The children also tired often and hard to awake. And also, the most common part is the children can experience a coma. Diagnosis of UCD. The diagnosis is made by the analysis of the urine and blood for abnormal metabolites, substance produced by metabolism, and high ammonia levels. A liver biopsy can be done to confirm the diagnosis, since it can show low level of enzyme activity. Genetic tests also can be done to show whether there is a problem with one of the genes needed to break down proteins of the urea cycle. To identify the particular type of urea cycle disorder can be treated through lifelong process that doesn't cure the condition, but it can effectively manage the symptoms. Frequently, blood tests are done to continue to monitor ammonia levels. Pediatrics, genetics, and nutritionists will work together to develop the child's or a person's treatment.
Urea cycle related disease in the form of hyperammonemia is used to illustrate a disease of metabolism specifically related to a defect in the urea cycle. A deficiency of one of the enzymes of the urea cycle leads to a specific disease condition where ammonia, which is a toxin, can accumulate in the blood. When glutamine is produced in excess in the liver, it is converted to ammonia by glutaminase. The liver urea cycle converts ammonia to urea that is exerted in the urine. And now that's all my part, and please listen to the next reporter. Thank you.
Good day to everyone. This is Marsha Barculia and I will be discussing the metabolic uses of acids. So, amino acid metabolism is an important process that occurs within the human body to assist in numerous biological reactions. And it is reprogrammed due to its important role in energy metabolism abnormality in tumor cells and are important processes such as the tissue growth, energy production, immune function, and nutrient absorption. Some of these metabolic uses of amino acids are the building block for protein synthesis, precursors of nucleotides and heme, source of energy, neurotransmitters, and precursors of neurotransmitters and hormones. We have seen before that during digestion in the gut, proteins are broken down into their constituent amino acids. Proteins contain 20 standard amino acids, which are incorporated into them during translation. Proteins may also contain several non-standard amino acids, so these are mostly formed by the post-translational modification and they are much less abundant than the standard amino acids. So, animal and humans obtain many important metabolites from their food and have a fairly lazy synthetic metabolism, while many other organisms, even simple ones such as the E. coli or the Escherichia coli, which are bacteria found in the environment, foods, and intestines of people and animals, can make all their amino acids from scratch. So we possess synthetic pathways for only about 11 out of 20 standard amino acids. So the remaining ones must be obtained from the diet, which is referred as the essential amino acids. Take note that protein is necessary and lacking it would lead to malnutrition. So most of the amino acids used in protein synthesis are obtained not just from food but rather through endogenous protein breakdown, which food protein only replaces the fraction of amino acids diverted toward their destinations. Now we have the outline of amino acid degradation. So first, the liver is the major site of the degradation for most amino acids, but muscle and kidney dominate the degradation of specific ones. So nitrogen is removed from the carbon skeleton and transferred to the acetylglutarate, which yields glutamate. So the carbon skeletons are converted into intermediates of the mainstream carbon oxidation pathways via specific adapter pathways. Then the surplus nitrogen is removed from the glutamate, incorporated into the urea, and excreted. So take note that all amino acids contain at least one nitrogen atom, which forms their A amino group. Several amino acids contain additional nitrogen atoms in their side chains. Some nitrogen is used in biosynthesis, that is the process which substrates are converted to a more complex products like the photosynthesis, amino acid synthesis, nucleic acid synthesis, and the ATP synthesis. So to this end, the liver incorporates into the urea, which is released into the bloodstream and is excreted by the kidneys. So the removal of nitrogen is typically an early step in the degradation and leaves behind the carbon skeleton. So the structure of the latter is different for each amino acids, and accordingly, each amino acid has its own specific pathways of degradation.
So the absence of key enzymes or imbalance in amino acid degradation leads to a severe disease states such as phenylketonuria, and methylmalonic aciduria.
So that is all for my part. Thank you for listening.
Hi, this is Carrie, and I'll be focusing on the amino acid breakdown pathways and the transamination of amino acids. So firstly, amino acid breakdown pathways join mainstream carbon utilization at different points of entry. Let's know more. Common amino acids are degraded by 20 different pathways, but the common skeleton converged them into 7 metabolic intermediate. These are the alpha-ketoglutarate, succinyl-CoA, fumarate, oxaloacetate, pyruvate, and the acetyl-CoA. So kindly observe the figure that I'll be showing here. The degradative pathways can be divided into 2 major classes. As shown here, most amino acids are converted to intermediates of the citric acid cycle or to pyruvate, which in turn can serve as precursors for gluconeogenesis. These are the glucogenic amino acids. Those amino acids that yield acetoacetate are called ketogenic. If you look closely, you will see the phenylalanine and the tyrosine are found on both sides of this division, since they yield both fumarate and acetoacetate. Some might say that that's what makes them both glucogenic and ketogenic, but others insist that either category should take precedence. Depending on the composition of our diet, amino acids may be very important as a source of energy. While plant-derived foodstuffs are typically rich in starch, meat high in protein but low in carbohydrates. Therefore, when on a diet that contains mostly meat, amino acids become more major source of glucose. What happens if protein is supplied in excess of the amount needed to cover our requirement for glucose? Well, the liver in principle contains enzyme activities required to oxidize the surplus carbon. Measurements of the liver's overall oxygen consumption indicate the most of the carbon that must be disposed elsewhere. Presumably, much of the carbon is still initially converted to glucose, which is then either stored as glycogen or released into the circulation. In the latter case, the surplus will mostly be converted to fat in adipose tissue. A surplus of ketogenic amino acid will mostly be converted into the liver to triacylglycerol. Let's talk about transamination of amino acids. Most standard amino acids lose their amino group early on the degradation through transamination. What is transamination? For instance, we have an alpha amino acid that we want to metabolize, and here we have a carrier molecule that accepts the nitrogen-containing group. So basically, the transamination is the process of an amino group, or is the process of the transfer of an amino group onto a different molecule. So when we transfer this amino group into a carrier molecule, we form our products. So almost always the carrier molecule that accepts the nitrogen-containing group is what we call as the alpha-ketoglutarate. After we transfer this amino group into the alpha-ketoglutarate, we form an alpha-keto acid and a glutamate. When we remove the nitrogen from the alpha amino acid, we form the alpha-keto acid. And when we give the amino group onto the ketoglutarate, we form glutamate. Now, when we form glutamate, what happens next will depend on the needs of the particular cell. So, when we need to form non-essential amino acids in that cell, then the amino group can be used by the glutamate to help form the non-essential amino acids. But if we don't need any amino acids, then the glutamate can then undergo the oxidative deamination. What's that step? It is the form of deamination that generates alpha keto acids and other oxidized products from the amino containing compounds and occurs primarily in the liver. So going back, the enzymes that catalyze this transamination step are called transaminase, which are also called as transferases. These enzymes are found in the cytosol of cells, and we find them predominantly in the cells of the liver, in the cells of the kidney, in the cells of the intestines, and in the cells of our muscle tissues. We have many types of aminotransferases depending on the individual amino acid that we're actually catalyzing. And the two aminotransferases that you have to be familiar with are the alanine aminotransferase and the aspartate aminotransferase. Let's talk about first the alanine aminotransferase, or the ALT. So from the word itself, alanine, it acts on the alanine amino acids. Here we have the alanine, which is our alpha amino acid, and the carrier molecule that accepts the amino group is the alpha ketoglutarate. So alanine aminotransferase catalyzes on the movement of the nitrogen-containing group from the alanine onto the alpha ketoglutarate. If we remove the amino group from alanine, we help form pyruvate, which is the alpha keto acid. But when this alpha ketoglutarate gains them in amino group, we form glutamate. Next is the aspartate aminotransferase, or the AST. We have exemptions to the rule, and this is one of them. Why? Because here we don't use alpha ketoglutarate as the carrier. Instead, we use another carrier molecule, namely the oxaloacetate. And the amino acid that we can act on is the glutamate. Aspartate aminotransferase catalyzes the movement of an alpha amino group. So the nitric-containing group from the glutamate to oxaloacetate, and so we form alpha ketoglutarate, and we also form aspartate. When we remove the alpha amino group from the glutamate, we form the alpha ketoglutarate. But when the nitric-containing group moves on the actual acetate, we form aspartate. The equilibrium of these reactions is 1. So we can go this way or we can go in reverse, and the direction that it follows depends on the conditions in the states within our cell. Now we have to remember that these aminotransferase, these enzymes require vitamin B6 as a coenzyme, and this is also known as a paradoxal phosphate. So these aminotransferase utilize vitamin B6, and so the paradoxal phosphate to actually function effectively and efficiently. The paradoxal phosphate is found attached covalently onto the lysine amino acid in the active site of these enzymes. And what this paradoxal phosphate does is it initially accepts this nitrogen-containing group from the alpha amino acid before it actually is transferred to the carrier molecule.
What is the fate of amino acid release in the protein digestion or turnover? Anything that is not needed as a building block or degraded into specific compounds. The major site of amino acid degradation in mammals is the liver. Amino acid must be removed so there will be no nitrogenous compounds in energy transduction pathways. The A fatty acids that results from the deamination of amino acids are metabolized so that the carbon skeleton can enter the metabolic mainstream. So therefore, the fate of A amino acids will be considered first, followed by the carbon skeleton. The A amino group of many amino acid is transferred to A ketoglutarate to form glutamate, which is then oxidatively deaminated to yield ammonium ion. Aminotransferases catalyze the transfer of an A amino group from an A amino acid to an A keto acid. These enzymes, also called transminases, generally funnel A amino groups from a variety of amino acid to A ketoglutarate to conversion of ammonium ion. Aspartate aminotransferase is one of the most important of these enzymes. It catalyzes the transfer of the amino group of aspartate to A ketoglutarate. Alanine aminotransferase catalyzes the transfer of amino group of alanine to A ketoglutarate. These transamination reactions are reversible and can thus be used to synthesize amino acid from A keto acids. The nitrogen atom that is transferred to A ketoglutarate in the transamination reaction is converted into free ammonium ion by oxidative deamination. This reaction is catalyzed by glutamate dehydrogenase. This enzyme is unusual in being able to utilize the nicotinamide adenine dinucleotide or nicotinamide adenine dinucleotide phosphate, at least in some species. The reaction proceeds by dehydrogenation of C-end bond, followed by the hydrolysis of the resulting Schiff base. Okay, so moving on, the equilibrium of this reaction favors glutamate. The reaction is driven by the consumption of ammonia. Glutamate dehydrogenase is located in mitochondria, as there are some of the other enzymes required for the production of urea. This compartmentalization sequesters free ammonia, which is toxic. In vertebrates, the activity of glutamate dehydrogenase is allosterically regulated. The enzyme consists of 6 identical subunits. Guanosine triphosphate and adenosine triphosphate are allosteric inhibitors, whereas guanosine diphosphate and adenosine diphosphate are allosteric activators. Hence, a lowering of energy charge accelerates the oxidation of amino acids. The sum of the reaction catalyzes the aminotransferases, and glutamate hydrogenase is the most terrestrial vertebrates. Ammonium ion is converted into urea, which is excreted.